Hello, myself Yukta Atle, and I will be talking about a research paper which tells us about isolation, characterization, and identification of antibiofouling metabolite from mangrove-derived Streptomyces Sampsonii PM33. Before knowing about antibiofouling metabolite, we should know about marine fouling. Marine fouling is a phenomenon in which deposition of marine life forms occurs on wetted surfaces like coastal regions or ships, etc. Fouling causes problems to shippings, aquaculture, and coastal industries. To stop this, synthetic or metal based antifouling agents, which are toxic or even lethal to non targeting marine life forms, were used or are being used. So, those researchers studied the microorganisms around the coastal area of India and they found out that Actinobacteria are the most common bacteria or microorganism around it. So actinobacteria are gram-positive bacteria having high G plus C content in their DNA. So they have unique potential in medicinal and pharmaceutical applications. It also has production of taxifolin. What is taxifolin? Taxifolin is a flavonoid which has antifouling potential. So the sample was isolated or collected from Vellaristorium, Parangapettai, Tamil Nadu, India. So this sample was then extracted using yeast extract, malt extract, agar surface fermentation. It was given for centrifugation, and after centrifugation, supernatant was tested against various solvents. Various solvents were chloroform extract, ethyl acetate extract, N-hexane extract, and methanol extract, in which ethyl acetate gave maximum amount of metabolite production. It means it is suitable solvent for production of that particular metabolite which shows antifouling properties. Then it was time for separation. Once we identified the molecule, we needed to separate each and every molecule to understand which molecule gives us maximum antifouling potential or activity. Thin layer chromatography technique was used to do so. Thin layer chromatography is a technique in which non-volatile mixtures are used to isolate. So the material usually used is aluminum oxide, cellulose or silica gel. Each component appears as spots separated vertically. Separation relies on relative affinity of compounds towards both the phases, that is stationary and mobile phases. Movement occurs in such a way that the compounds which have higher affinity to the stationary phase move slowly while other compounds travel faster. So, this tells us about the individual components from mixture which appear as spots. Then, spot 2, that is second spot, was identified as maximum inhibitory organism against Staphylococcus species M1 and it was named as PM33. Also in TLC, solvent system used was N-hexane equal to ethyl acetate. So in total, 125 milligram of active PM33 was obtained from 1 gram of crude extract. Then techniques used for identifying the physical or chemical properties of the compounds are as follows. UV visible spectrophotometry, FTIR analysis, NMR analysis, and MS analysis. UV visible spectrophotometry. It is based on interaction between light and matter. When light passes through or is absorbed by a molecule, it can cause the molecule to vibrate. The wavelength of light that is most strongly absorbed by a molecule is called absorption maxima. By measuring the absorbance of light at different wavelengths, it is possible to identify and characterize the molecules. Next is Fourier transform infrared spectroscopy. The goal of FTIR is to measure how much light a sample absorbs at each wavelength. So rather than shining a monochromatic beam of light composed of only single wavelength at the sample, this technique shines a beam containing many frequencies of light at once and measures how much of that beam is absorbed by the sample. Next, the beam is modified to contain a different combination of frequencies, giving a second data point. This process is rapidly repeated many times over a short period of time, and afterwards, a computer takes all this data and works backward to infer what the absorption is at each wavelength. Next is NMR technique, that is nuclear magnetic resonance. So many nuclei have a spin, and all nuclei are electrically charged according to the NMR principle. An energy transfer from the base energy to a high energy level is achievable when an external magnetic field is supplied. So, this NMR technique is used to provide reaction state, structure, chemical environment, and dynamics of molecule. Then, mass spectrometry. Mass spectrometry is based on Newton's second law of motion and momentum. A mass spectrometer uses this property of matter to plot ions of varying masses on a mass spectrum. From the law, we infer how much mass is relevant to the inertia and acceleration of body. This principle is applied to the aspect where ions with different mass to charge ratios are deflected by different angles in an electric or magnetic field. We have results. Results state that Streptomyces Sampsonii PM33 was this bacteria which can inhibit the activity of biofouling organisms. Its molecular formula is C15H12O7. Properties shows us that PM33 strain is to be a taxifolin. Antifouling assay. Antifouling assay states that purified taxifolin showed antifouling activity against biofouling bacteria, and it reduced 70% of algal spore germination. Some mollusks were also dead in the test tubes due to effect of taxifolin. Taxifolin applied on wooden boat showed its effect, and there was less settlement of biofouling organisms after 2 weeks in comparison to the controls. The PVC panels had a small covering of biofouling organisms even after 4 weeks. Next is toxicity. Toxicity testing was done in zebrafish model. Zebrafish embryos treated with taxifolin had pericardial edema around their heart. So after increasing the concentration of taxifolin, more embryos had pericardial edema. This states that taxifolin possesses less toxic effects in lower concentration only. Conclusion of this study is that Streptomyces species can produce taxifolin which has antibiofouling activity or properties. Taxifolin inhibited algal spores and attachment of mollusks. Taxifolin could be eco-friendly as it is not harming any other non-targeted marine life forms and is not spreading any kind of toxicity. We can suggest that additional field studies are to be designed in future to check the effectiveness of taxifolin on marine life to support taxifolin as a replacement for chemical antifoulant. So this was the explanation from my side for the research paper. Thank you.